Hello and welcome to this Introduction to Microbiology video. I'm Dr. Aled Roberts, and in this video we're going to look at the basics of chemotaxis and how this affects flagella swimming in bacteria. Now, the general term for bacteria moving in response to some form of external signal is taxis, and this movement or taxis can be either positive or negative. And so a positive taxis means the organism will move towards the stimuli, whereas a negative taxis means the organism will move away from the stimuli. So as an example, a bacteria might want to move towards nutrients and move away from something like antibiotics. Now, there are many different types of stimuli that affect our bacterial cells. So bacterial cells can respond to light, and we call these phototaxis. They can respond to water, and we call this hydrotaxis. They can respond to oxygen concentrations, and we call this aerotaxis. Now, one of the more common taxis out there is what we call chemotaxis, and this is where our cells respond to chemicals, and we'll look at this in more detail in a minute. But before that, just some other less well-known taxis. We have cells that can respond to electric fields, known as galvanotaxis, magnetic fields, known as magnetotaxis, and ionic strengths, known as osmotaxis. Now, in each of these instances, there are two sides to the stimulus. We have attractants and repellents. Attractants are favourable environmental signals that bacteria move toward, while repellents are harmful or stressful signals that they move away from. These signals are detected by receptors which activate a signalling pathway that controls flagella movement towards attractants or away from repellents, allowing bacteria to navigate their environment for survival. Now, for the rest of this video, we are going to focus on chemotaxis, but the general mechanism is similar for each. And so in its basic form, chemotaxis is the movement of an organism in response to some form of chemical stimuli. And this can be in the form of a chemoattractant, which includes sugars such as glucose, maltose and ribose, amino acids such as aspartate and serine, or various elements such as ion complexes that some bacteria require for survival in a host. On the flip side, we also have chemorepellents, which include various toxic substances such as phenols and organic acids, antimicrobial agents such as tetracycline and host-derived defensins, and some more general stressors such as salts, high pH environments, and the presence of metabolic byproducts. Now, the taxis systems, particularly the chemotaxis system, is linked to the bacterial flagella, and it is the flagella that generates the motion required to move closer to or further from the stimuli. Now, the flagella has two main forms of movement. It can rotate in such a way to promote free swimming in a single direction, or it can rotate in such a way that the cell reorientates, with both being required for the process of chemotaxis. And if we were to look at this in more detail, when a chemoattractant is present, the flagella rotates counterclockwise. Now, the key thing here is that due to the helical construction of the flagella filament, counterclockwise rotations allow multiple flagella filaments, if present, to align into a cohesive bundle, as we can see on the screen here, and this propels the cell forward. Now, we call this movement free swimming, or a run as it's more commonly referred to. So that's our first type of movement. The second is when a chemoattractant is absent or a chemorepellent is present. Now, in this situation, the flagella will rotate clockwise. Now again, due to the helical construction of the flagella filament, a clockwise rotation disrupts that cohesive flagella bundle, causing the flagella to separate, as we can see on the screen here. And naturally, this causes a loss of propulsion. Now, the splaying of the flagella generates some chaotic forces on the cell, resulting in what we call a tumbling motion, where the cell reorientates itself, hopefully in a more favourable direction. Now, you might assume that runs are good and tumbles are bad. However, it's the alternation between runs and tumbles that results in a random pattern of movement, which is called a random walk. Now, this random walk isn't entirely aimless. It is somewhat biased by environmental signals. So what do I mean by this? Well, when a bacteria detects an attractant, it reduces the frequency of its tumbles, favouring longer runs in the direction of the attractant. And then on the flip side, if it senses a repellent, it shortens its runs, increasing the likelihood of tumbling and reorientating itself away from the repellent. And so this bias transforms the random walk into what we call a biased random walk, allowing bacteria to move towards favourable conditions and away from harmful ones. And we can visualise this as we can see here. The cell will move forward until it detects a repellent, and then it will tumble. It will reorientate itself and move forward. When it tumbles again, moves forward, tumbles, moves forward, tumbles. Each time it tumbles, it's reorientating itself, and hopefully over time it will slowly make its way towards an attractant. Now all of this is made possible because of sensor proteins embedded in the bacterial envelope, and these are known as chemoreceptors. And they are essentially transmembrane proteins that detect chemical signals, such as our chemoattractants or chemorepellents outside of the cell, internalizing this as a signal cascade so the bacteria can act upon it, which in this instance is whether the flagella needs to rotate clockwise or counterclockwise. And so we're going to have a little bit of a look at this signalling process, starting with the absence of our chemoattractants, and again we're going to use E. coli as our model organism. So within the inner cell membrane, usually located towards the poles of the cell, there is a chemosensory complex made up of methyl-accepting chemotaxis proteins, or MCPs for short. CheW is also attached to this, along with CheA. And so the MCPs act as a transmembrane receptor that senses attractants and repellents. CheW helps signal transduction between the MCPs and CheA, which is a kinase protein that autophosphorylates, meaning it can gain a phosphate group and can transfer this to downstream response regulator proteins. Now in its current state, as we can see here, the conformation of the proteins puts it in an active signaling state, meaning it promotes the autophosphorylation of CheA. Now to do this, ATP is used as a source of phosphate groups and is broken down into ADP, with CheA retaining that terminal gamma phosphate group. And now in its active phosphorylated form, CheA is able to participate in a number of reactions, transferring the phosphate group to different downstream response regulators, a response which is now linked to the absence of attractants. Now one of the first response regulators we're going to look at is CheY. Now CheY is found throughout the cytoplasm of the cell, but when in close proximity to an activated CheA protein, it can become phosphorylated, putting it into an activated state. Now the concentration of phosphorylated or activated CheY will be higher around chemosensory complexes in this active signaling state, creating a spatial concentration gradient, which essentially means you will get higher concentrations of phosphorylated CheY near the activated CheA chemosensory complexes. And this concentration will gradually decrease as it diffuses away from the source. More on this in a minute. Okay, so what does our activated CheY protein do? Well, its core function is to interact with the bacterial flagella, specifically the FLIM protein located within the C ring. Now this interaction is critical because FLIM acts as a switch to determine the direction of the flagellar rotation. When phosphorylated CheY binds to FLIM, it triggers a conformational change within the C ring, which causes the motor to shift to clockwise rotation. And hopefully you will remember that this disrupts the smooth bundle of flagella responsible for free swimming and causes the cell to tumble and reorientate itself, changing direction. Now if you remember, I said that the concentration of phosphorylated CheY decreases the further you get from the chemosensory complex. Well, that is due to the presence of CheZ. Now, CheZ is an essential phosphatase in the bacterial chemotaxis system and is responsible for resetting the signalling pathway so that the cell doesn't constantly tumble through the clockwise rotation of the flagella. Now, it does this by dephosphorylating activated CheY into its non-active unphosphorylated form, and so by controlling the life cycle of phosphorylated and non-phosphorylated CheY, the cell can ensure a rapid and reversible response to environmental signals. Now, if we take 2 steps back and if we look at the chemosensory complex in its active signalling state, it is able to interact with a second response regulator, CheB. Now, CheB localizes around these chemosensory complexes, and when in close proximity to an activated CheA protein, it can become phosphorylated, putting it into an activated state. Now, CheB plays a role in adaptation, and what I mean by this is that it influences the sensitivity of the MCPs to attractant and repellent signals, helping the cell adjust its response over time. And it does this by removing methyl groups from specific glutamate residues on the MCPs, but can only do this when it's in its activated phosphorylated form, essentially acting as a methyl transferase. And so by removing the methyl groups, it makes MCPs more sensitive to attractants and less sensitive to repellents, which will ultimately cause a switch to an inactive signalling state. Now, obviously, before we get demethylation, we need to have methylation, but we'll go through that in just a minute as we now turn our attention to when chemoattractants are present. And so, if a bacteria finds itself in an environment filled with attractants, they will be taken into the periplasm and accumulate. If concentrations are high enough, they will bind to methyl-accepting chemotaxis proteins, and in doing so, they will cause a conformational change throughout the chemosensory complex. This leads to the complex switching to a sort of inactive signalling state. And what this essentially means is that CheA is now unable to phosphorylate, which means that there is no source of phosphate groups for downstream signalling cascades. Therefore, CheY doesn't become phosphorylated. And if CheY doesn't become phosphorylated, it's not activated and it is unable to fully interact with the FLIM protein of the C ring. Now, when FLIM isn't bound to anything, the C ring is in its standard state, which causes the motor to turn the flagella in a counterclockwise rotation. And hopefully you'll remember that this allows the flagella around the cell to form a bundle that propels the cell through the environment in a free-swimming motion, directing it, hopefully, towards the attractant. Now again, if we take 2 steps back, the chemosensory complex in its inactive signalling state is able to interact with a protein known as CheR. Now, CheR is a methyl transferase that plays a role in adaptation, and what we mean by this is that it influences the sensitivity of the MCP to attractant and repellent signals, helping the cell adjust its response over time. Now, it does this by adding methyl groups to specific glutamate residues on the MCPs, and I know it's taken a while to get here, but this is where the MCP gets its name as a methyl-accepting chemotaxis protein. And so the addition of these methyl groups make MCPs less sensitive to attractants and more sensitive to repellents, which will ultimately cause a switch back to the active signalling state. And that essentially wraps up the process of how bacteria sense stimuli and respond through rotational changes within the flagella. And as I said earlier, whilst we've only looked at chemotaxis and chemical stimuli, the fundamentals are very similar in other taxis systems, allowing bacteria to move towards a range of attractants and away from repellents. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.